membrane, and here's photosystem 2 and photosystem 1. You can plan for this. Here's oxidative phosphorylation. Both of those can provide energy that's needed to open closed stomata. Here's the sequence of events. Somehow a signal comes in, like the lights turn on. Somehow the guard senses it, and I'll get into that sensing mechanism later. But first you have an environmental signal that's perceived. The first thing that happens is the guard cells pump protons out of the guard cell into the apoplast, across the plasma membrane. So protons come from inside the cell into the apoplast. That creates a pH gradient across the plasma membrane and an electrical potential difference across that membrane because you're pumping a positive charge from inside to the outside. So that pH gradient, electrical potential gradient is set up by pumping the proton. And this proton extrusion is what requires energy. Remember in the phloem loading that, no, that's on here, yeah, that's on here. Oh, no, that's not on here. That, what's it called? The protein complex in the membrane that takes ATP and makes ADP out and gets the energy and pumps the proton. That thing, that requires the energy and it pumps the proton. Anyway, that sets up a electrical potential across the membrane. When that electrical potential becomes high enough, so it's positive out and negative in, the positive potassium ion can move passively in response to that difference in electrical potential, right? It's positive, it's going to move to the negative thing like a magnet. So the potassium enters passively. Chloride, though, being negatively charged, is likely taken up actively, since that movement is against its potential, its concentration gradient and its membrane potential. Right? So once potassium and chloride concentration increases in the cell, the total water potential, that makes the osmotic potential more and more negative, causing that guard cell to be in a disequilibrium with the total water potential around it. And water moves in from the surroundings into the cell, increasing its volume and equilibrating its water potential. As the cell volume grows, the cells distort and the thing opens up. But it's all characterized by this first pumping of protons across the plasma membrane from inside the guard cell to out in response to some environmental or hormonal signal. All right, now, when protons leave the cell, when protons are pumped out of the cell from in response to this environmental signal, the cytoplasmic pH increases. Right? The pH gets higher and higher and higher. And the guard cell cytoplasm not being buffered well, it needs another mechanism to regulate its pH, right? Cells like to regulate their pH and have it within certain boundaries because enzymes, lots of enzymes are active or inactive depending on what the pH is. So living cells tend to regulate their pH. Now, most cells regulate their pH by pumping protons across the membrane when they need to. But guard cells are doing that to open and close, and so they can't use that to regulate their pH. They likely regulate their pH through the production and degradation of organic anions like malate. And so that's why that concentration varies a lot when the cell is open versus when it's closed. And it's kind of a contrast to a lot of other cells, but that's the way it works. Okay, so those are three factors that change quite a bit, or three substances, three solutes that change, whose concentration changes: potassium chloride and malate, organic ion. Remember, malate is a 4-carbon compound. All right, here's a great figure, believe it or not, from Tyson Zeiger's book, Plant Physiology. And it shows different osmoregulatory pathways that happen in guard cells when closed stomata open. And the big arrows are the main ones, the smaller arrows are the ancillary ones. Okay, so in this case, what do we have? Oh yeah, potassium and its counterion. In the chloroplast in the guard cell, by doing photosynthesis, this 3-carbon compound can be exported. And lots of cells will incorporate this into sucrose in what we call in the whole process of photosynthesis. But in guard cells, this 3-carbon compound can be carboxylated to make malate. And that concentration changes a lot. So the carbon in this malate can come from current photosynthesis via the Calvin cycle, or from degradation of starch in the same way. And the malate regulates the cell pH, which is changing because the cell is pumping protons out and potassium chloride are coming in. On the other hand, carbon from current photosynthesis or from degradation of starch in the chloroplast can export the leave the chloroplast in the form of this 3-carbon compound and be incorporated into sucrose. We'll see in a minute, sucrose concentration also changes a lot, but on a slower time scale. Also changes, just like chloride and potassium. On the other hand, this sucrose inside the guard cell likely comes from current photosynthesis. This is what a lot of cells do, this part, without this stuff on it. This is the Calvin cycle, and this is sucrose synthesis. If you look over the course of a day, here we see stomatal aperture. And here's the time of day. So that's these bodies. So early in the morning, stomata fairly closed, the sun comes up, it gets lighter, stomata open up, midday, and then later in the day as the sun goes down, they close again. That's the behavior. If you look at potassium concentration, that's this series of open symbols. And when the guard cells when the stomata are opening, the potassium concentration tracks that opening very well. So potassium is accumulated. But after a while, the potassium concentration in the guard cells decreases. But they're still opening and remaining open, even though the potassium concentration subsequently declines in these cells. If you look at the sucrose production, when the guard cells open and the potassium accumulates, eventually the sucrose concentration gets high. As the potassium concentration decreases, sucrose concentrations are increasing. That keeps the osmotic potential low in the guard cells and keeps their pressure potential high, their volume high, and the pore open. Later in the day, when they close, the sucrose concentration also decreases. So sucrose is important too, but its effects are shifted a little bit relative to potassium ion. And that's what Theis and Zeiger are trying to incorporate here. You know, the guard cells have all these pathways for making various ions. That's what's happening with the guard cell metabolism. Next, on Tuesday, what I'd like to talk about are specific environmental cues that regulate stomach conductance. I'll start off with arguably the most important one, light. How is it that the guard cell perceives light and then subsequently causes that extrusion of protons that sets up this whole scheme of ionic changes that keep the stomach pores open? So that's where we'll go. Have a good weekend. See you all on Tuesday.